I am Dr. Shija. Today again I am with my new lecture number 10 of General Virology, which is on single-stranded RNA viruses. So our focus is HCV virus, hepatitis C virus. So these are the contents of my lecture today. So first of all, that is the introduction to family Gemini viridae, genera in this family. Then we will study about the HCV virus, which belong to the genera Hep C virus. Then we will study about the genome of HCV virus. Then we will know about the transmission routes of HCV. Then followed by the life cycle and worldwide the prevalence of HCV virus. And in the last, we will focus on the significance of HCV virus. Before starting the lecture, let me give a brief introduction of family Flaviviridae. So this family belong to the viruses which infect human beings and other mammals, and they are served as a natural host for this virus. In this family, mostly the viruses are vector-based, are transmitted by the arthropod vectors, including ticks and mosquitoes. This family gets its name from the yellow fever, and this type virus of Flaviviridae. Flavus means yellow in Latin, and yellow fever in turn was named because of its propensity to cause jaundice in victims. There are more than 100 species are included in this family, which have been divided among 4 genera. So the main genera of this family are Hep C virus, in which is the main prototype is the hepatitis C virus, then followed by the pesty viruses and flaviviruses. Dengue virus also belong to this family, and in the last one is the PEGI virus. There is not a lot of information has been reported about the PEGI genera. Our main focus is the hepatitis Hep C virus of this family. These are the general characteristics of family Flaviviridae. So if we are talking about the genome architecture, so virus genome of the family Flaviviridae are single-stranded positive sense non-segmented RNA of approximately 9 to 11 or 13 kb. So if we are talking about the flavivirus, so its genome size is around about 9.2 kb to 11 kb, festivirus 12.3 to 13 kb, hepativirus start from 8.9 kb to 10.5 kb, and pegivirus 8.9 to 11.3 kb respectively. So the virion contain only a single-stranded long ORF flanked by 5' and 3'. So this indicates that this is a linear RNA because there are no 5' or 3' in a circular genome. So on both sides there are UTR untranslated region or non-coding region which form a specific secondary structure and responsible for some sort of enzymatic activities and biological activities and play a vital role in replication and translation. So in some of the viruses in this family are cap dependent. In case of members of the genus flaviviruses, dengue virus belong to this genera. So this is the genomic architecture which containing the structural and non-structural genes. The structural genes round about are 3. Encode for the core protein envelope 1 and the membrane proteins are in the envelope 2. And the non-structural proteins has been divided into the NS1 to NS5 and responsible for different sort of activities. So now let's focus on the genus Hep C virus. Is a viral infection of liver caused by the hepatitis C virus. So this virus is basically a global burden of hepatitis C virus, which is responsible for a major public health problem worldwide. Around 180 million people are infected with the hepatitis C virus. It accounts for 3% of global population. Just imagine the importance of this virus. And 3 to 4 million people become infected with hepatitis C virus annually. So this virus is the basic causative agent of hepatitis C in humans. and this is a member of the Hepatitis C virus which was discovered in 1989. There are 7 genotypes of hepatitis C, and there are further division or categories of each genotype, which we can say that sub genotypes or subtypes of each genotypes, and they are usually dominated or presented with the small letter of English. For example, genotype 1 has 3 or 4 subgenotypes, so each subgenotype has been presented as 1A, 1B, 1C like this of hepatitis C virus, and these are distribution has been done for the understanding. So 1A and 1B is globally distributed and prevalent in the globally prevalent, while if we are talking about Pakistan, so the subgenotype 3A is predominant. HCV is more dangerous than HIV because the number of patients of HCV are more than HIV or AIDS. Hepatitis C is the cause of hepatitis C, and some cancers such as which are finally the infection in the liver are finally developed into some sort of cancer. For example, hepatocellular carcinoma abbreviated by the HCC are lymphomas in humans. If we are talking the virion of HCV, so the virion size around about 40 to 60 nanometer in diameter, spherical in shape, and contain a lipid envelope, and they HCV virus are mostly transmitted vertically or horizontally. So we will talk about the transmission of HCV virus in the coming slides. The genome of HCV virus is a positive single-stranded RNA genome. Already I have explained about this, which and it composed of 9.6 base pair nucleotides, and which encode for only a single proteins known as polyprotein, which are further processed into different protein structure and non-structural proteins. The single open read frame is translated to produce a single protein product, which is further processed already explained. So at the 5 prime and the 3 prime of the RNA are the UTR or non-coding regions, untranslated regions that are not translated into any product or any proteins, but still no one can neglect the importance of these structures because they play a vital role in translation and replications of the viral RNA. At the 5 UTR of the HCV genome, there are ribosome binding site or entry sites. So itself are not translated but involved in the ribosome entry sites. Means translation will take place from this region, and the polyprotein are produced, so that is around about 3000 amino acid. So these are the further information about. So there are around about 10 open reading frames are encoded for the structural proteins, and the rest are all responsible for the non-structural proteins. So the pre-large protein is later cleared by the cellular and viral proteases into 10 smaller proteins that allow viral replication within the host cells are assembled into mature viral particles. So the structural proteins include the core protein, envelope 1 and envelope 2 glycoproteins, while the non-structural proteins are the functional protein include NS1, NS2, NS3 up to NS5, which is responsible for the which encode for the RNA-dependent RNA polymerases. So this is the basic genomic organization of hepatitis C virus. So on the 5 prime we can see the untranslated region which produce are not like shape. This is known as the soldier knot, and it play. So these structures, secondary and tertiary structure, are responsible for some enzymatic activities or biological activities, and it provide ribosome entry sites, and then the core protein or capsid protein followed by the E1 E2. These three are the non-structural proteins, and the rest of all from NS1 to NS2, NS3, and up to NS5B are responsible for the structural proteins and play different sort of functions: protease activities, helicase activities, like regulate replication and virus assembly, and the last one is the most important, that is known as the RNA dependent RNA polymerases, which produce the negative strand of this virus and play important role, and that negative strand is used for the as a template for the production or the construction of positive strands. And at the 3 there is 3 prime, there is again there is untranslated region. So the 3 UTR and 5 UTR are not a part of any coding region but still involved in the replication and other activities. If we are talking about the transmission of HCV virus, so HCV virus can be transmitted by vertical or horizontal. Vertical means from parents to offspring, and we are talking about the horizontal, so from one person to another through surgical equipments, needles, tattoos. Sometimes we are going to hospitals, some for beautification we are making tattoos. The needles are contaminated, are not properly sterilized, so we are going for beautification to some dental hospitals. Dentistry play a vital role, and sometimes we are visiting the barber shops. So all these things are even drugs, contaminated drug user. So virus transmitted by this is known as the vertical transmission of the virus. Uh, the virus can also be transmitted sexually. If a mother who is infected with, there are more chances the newborn baby will be also positive for the HCV. It should be noted that HCV virus is not transmitted by air, droplets, vectors, or from animals, and there is no any sort of report in the database about these things. If we are talking about the life cycle of HCV virus, so it life cycle can be easily understood by dividing the whole mechanism into different steps: attachment, entry, uncoating, translation, and so going on. So the virus replicates mainly in the hepatocytes of the liver, where it is estimated that daily each infected cell produces approximately 50 virions. Yeah, virions are the functional virus or a complete virus which is responsible or can infect any cells. So the virus may also replicate in peripheral blood mononuclear cells, potentially accounting for the high level of immunological disorder found in chronically infected HCV patients. So because HCV doesn't replicate through a DNA intermediate, so it doesn't integrate into the host genome, because this virus replicate in the cytoplasm, not in the nucleus. And this virus is RNA based, is a as a positive configuration, and positive configuration means a messenger RNA directly loaded on the ribosome of the host cell and translated into protein. And some viruses are replicate by producing replicative forms, double-stranded DNA or cDNA. So there are more and more chances any virus in a double form, a replicative form, integrate into the genome of the host cell, like HIV virus. So HCV is never integrated, and there is no any report to be integrated to the host genome. So for the attachment or absorption, HCV virus always enter into the host cell by attachment. The E1 and E2 protein recognize and bond with the CD8 receptor present on the surface of hepatocytes and lymphocytes. After the interaction of the virus envelope with the host cell membrane, HCV enter the cell through endocytosis. In the cytoplasm, the messenger RNA then undergoes translation and polyproteins are produced. So, attachment is totally based on specific receptors, and these receptors are only present on the liver cells. That's why HCV virus are specific to liver cells. For entry after attachment, then the entry. And how do its entry? Entry is only possible in the form of cytosis, so it's move inside. This indicates endocytosis, and this always takes place through erythrin mediated endocytosis. Now the virus is present inside the liver cell. Next step is the uncoating. How uncoating will be take place? It will be done by the pH of the endosomes, low pH, which do some sort of conformation, and the RNA is released inside the cytoplasm. Now, the naked RNA, which is available in the cytoplasm, has the potential to load on the ribosome of the host cell for translation, because RNA has the potential for serve as a messenger RNA. Now it will follow the rule and regulation of replications. For replication, there is a protein known as the RNA-dependent RNA polymerase, which will produce the negative strand of the messenger RNA, which is a positive strand, and that will be used to produce more and more messenger RNAs. Finally, assembly and maturation take place. After replication, the HCV RNA then replicates, after which the new viral RNAs are packed and transported to the surface of the host cell so that they can disseminate and complete a new cycle. So now proteins are available, RNAs are available, RNA will go inside the proteins and they will be released in the form of budding, which is known as the exocytosis. So here we can see the life cycle of HCV. In the first, it do the entry. Entry is done through attachment, endocytosis, and this is clarity mediated endocytosis. Then followed by the uncoating. So here is replication and translations going on and on both sides. Then finally is the assembly and maturation, and finally the virus virions are released in the form of budding to attack a new cells or reinfect the same cell.